Hi guys, welcome to our video. Today we're going to be talking to you about lichen, but we'd like you to join us. I will be your tour guide with my lovely partner Annabelle. My name is Grace, by the way, and we hope you get really excited and passionate about lichen today.
A lichen is not really a plant. It's not really a fungus either. It consists of many different organisms living together that are heterotrophs and phototrophs. The phototrophs are mostly algae. They live together and together they look like that. Like kind of like a fungus, but not really. This is Ivernia.
This is Ramalina.
We collected samples of lichen in 3 different study locations in the South Bay area near San Francisco. The 3 locations are Jasper Ridge Biological Preserve in the northwest, Arrastradero Nature Preserve in the south, and several smaller sites around the campus of Stanford University in the east. Every location consisted of a number of study sites that all followed the same protocol. This is one of our study sites where we collect all our data. The pink flag marks the site where we want to collect the lichen, and we usually start off with finding lichen that are infected and uninfected to estimate their age and their abundance at the site. And then we collect some samples that we can bring back to the field preserve or the lab to further assay. Okay, this Ivernia, this branch had 4 Ivernia and was 20 centimeters long.
At every study site, we collected 4 lichen total, 2 infected and 2 uninfected ones. They would later be used for growth experiments and genome sequencing in the lab. To examine air pollution at our sites, we collected another 10 Ivernia prunostri lichen that will be sent to the stable isotope lab to be analyzed for their content of air pollutants like nitrogen. In the lab, we did several experiments to analyze our lichen samples. We put a few of them into a growth chamber for a week to estimate their growth rate. After the growth experiment, we measured the photosynthetic rate of the lichen. What you see us doing here is using a machine called a mini PAM2 to measure the photosynthetic capacity of our Ivernia samples. This is a metric that we will use to look at the fitness of our lichen samples.
In our first analysis, we looked at how nitrogen content and age of the lichen influenced their infection status. Ivernia lichen are regularly infected with the parasitic fungus Unguculariopsis lettaui, or short, young Larry. We found that where nitrogen has no effect on susceptibility to be infected, older lichen were a lot more likely to be infected than younger ones. We explained the results like this. The longer the lichens are around, the more likely it is that they will get infected at some time. Older lichen could also be weaker and less healthy to begin with, and are just more susceptible in general.
For our second analysis, we wanted to know if nitrogen content or age has an effect on the photosynthetic capacity of the lichen. We did not find any significant effect for either of them until we divided our lichen into age groups. For the oldest age group, which are lichens that are from 7 to 18 years old, lichens with a higher nitrogen content showed an increased photosynthetic capacity, which we use as a measurement of fitness. This was surprising because we expected lichen to decrease in fitness with higher air pollution, but nitrogen can also act as a fertilizer, so maybe nitrogen content at our study sites did not reach a detrimental level for the lichen and instead boosted their fitness. Lastly, we also looked at the effect of parasitic infection on photosynthetic capacity or fitness. We saw that whether the lichen were infected or not did not really have an impact on photosynthetic capacity. So we concluded that young Larry infection does not really affect the lichen's fitness. This type of interaction between fungus and the lichen depends on the environmental conditions for the lichen. Therefore, this type of symbiosis could be mutualistic rather than parasitic because of the conditions, and that they're maybe not ideal for the lichen. Overall, age seemed to play a major role for our lichen. Older lichen are more likely to be infected, and for older lichen, higher nitrogen content influences their photosynthetic capacity. Interestingly, infection by a parasitic fungus did not influence our metric of fitness, but that does not mean other metrics of fitness are not affected by parasites. Nitrogen content did not negatively impact any metric that we used on our lichen, even though we predicted air pollution to be damaging to the lichen. Maybe Ivernia prunostri are more resilient than we thought. Hi guys, welcome to the end of our hike. So Annabelle and I are out here today at the Stanford Dish collecting our last couple lichen samples. We hope you've learned a lot on the journey and we're happy to answer any questions you might have about lichen. Thanks for joining us.